In section 3 of chapter 11, we'll be looking at a little bit of details on how neurons function, which is called electrophysiology. And so just as a review, we already know some things about neurons. So neurons are excitable cells. That just means that they have the ability to respond to different signals or different stimuli. It could be a chemical, it could be another action potential. It could be some kind of mechanical stimulus as well. And we'll see an example of that a little later on. Once they become stimulated, then they have the ability to transmit those electrical charges across membranes, and those action potentials we've seen before in the muscle chapter. We'll kind of review that briefly here in chapter 11 as well. And they have the ability to conduct those action potentials along the length of the axon, along the length of the neuron's membrane. So they have the property of conductivity. And so generally when you talk about electrical changes in neurons, you think of either localized potentials, which is just a potential that travels a very short distance, maybe just within the neuron or down the length of the axon. But if you're talking about an action potential, technically that includes the entire axon, the whole length of it if it branches, but the entire length of axon would be what we refer to as an action potential. Now, a couple things about how these action potentials are being sent, or the speed of these action potentials. Generally, you find action potentials going much faster in the axons that are myelinated. In the neurons that are myelinated, they have the myelin sheath, you know, the Schwann cells and the oligodendrocytes that form the myelin sheath. That allows for action potentials to propagate or to move through the axon much quicker. And generally, the thicker that myelin sheath is, the faster the speed of conduction. Also, the larger the axon is, the faster it's able to transmit that action potential. And so I found this diagram that kind of shows how action potentials travel down the length of an axon. It's a wave-like action almost. It's a propagation type action. And you can kind of go through the steps here, but what you'll see is once one section of the axon becomes depolarized, and so you see a depolarization event taking place here, that in turn triggers the neighboring portion of the membrane to become depolarized, and then that kind of piggybacks on that. It's like a domino effect, and eventually that action potential is propagated down the length of the axon. This is referred to as continuous conduction. Now that's a little different than something called saltatory conduction, and that's where the action potential actually is generated only in places where you have these nodes. We mentioned before you have something called a node of Ranvier. The place where you have the Schwann cell, the myelin sheath, that's the internode, and then the node area is really the only place you have to have a new action potential generated. And so what that means is if you have the Schwann cells in place, you have the myelin sheath in place, that allows the conduction of that action potential much faster, to go much faster. And so that's because it's able to go through the axon here without having to, change the membrane potential each time all along the axon. It only has to change it at the nodes. And so that speeds up. So that's kind of helps to explain why myelinated axons are much faster in transmitting those action potentials. Now, to help generate those action potentials, you have to have different types of channels in the membrane, and this is very similar to what we saw in muscle cell physiology. You've got different types of channels. We'll look at these, kind of review these. You have leak channels, and then some of them are gated channels. And if they're gated, they can either be ligand gated, voltage gated, or some other types of mechanical gated situations. So let's kind of review some of these. Leak channels or leak ion channels. A couple of properties here. They're always open. So in other words, they're non-gated. They don't have really a control mechanism other than they're maintained by diffusion. So they have to operate based on the laws of diffusion, which says that molecules or ions in this case will go from high concentration to low concentration. Now they generally are specific for one particular type of ion. And one of the main leak channels that we've looked at before is potassium. Potassium is a leak channel, although there are sodium channels, sodium leak channels. and even chloride. And you kind of see in this diagram, you even have a chloride type channel here. A lot of times chloride channels are voltage gated, but here you have some mainly sodium channels that are leak gated, and then also potassium, and that allows these ions to move back and forth across the membrane through their concentration gradient. Now, to keep everything in check, though, to keep everything in balance, you have to have a way to push these ions back across, and so that's why you would have a sodium-potassium type pump mechanism to try to build up the concentration on either side of that membrane. Ligand-gated channels. A ligand is basically just another name for a molecule or a chemical, and in this case, these are gated channels. They're usually closed until they have a ligand present, some kind of molecule present. And so once that molecule binds to the channel, that opens the gate. And then you have the ability for ions to move through that ligand channel. A lot of times sodium channels are ligand gated. You have to have a molecule. Many times, one example is the neurotransmitter acetylcholine. Once you have acetylcholine in place, and a lot of times you find that in a synapse between a neuron and a muscle cell, once you have the acetylcholine in place and it binds to its receptor on a sodium gated channel, that causes that sodium gate to open. The channel opens, and that allows the sodium to go through, and basically you're transferring that action potential from the end of the neuron cell to the muscle cell. So that is a ligand gated channel. You also have voltage gated channels. You see this pretty frequently. This just means that a channel opens in response to, another action potential, a small voltage change, which is electrical signal or action potential, however you want to call that. It's all kind of the same thing, but a small change in voltage causes that gate to open. And you see these a lot of times, especially for sodium channels, but there's also voltage gated potassium channels as well. Then just like we looked at in the muscle cell, you'd be able to measure the membrane potential of a neuron, just like you can for a muscle cell. And this example here is kind of showing you a couple things to review. Here you have the plasma membrane. This is a lipid bilayer. Usually you have more negative ions on the inside of the cell, and more positive ions on the outside of the cell. Now this is at resting membrane potential. And so if you could put a probe into the cell, you would measure the inside somewhere around -70. Sometimes it goes up to -90 millivolts on the inside of the cell. And remember, a change in the polarity here because of a gate opening will cause a change in the polarity. That gives you your action potential. Now, there are some other gated ion channels I mentioned before, such as touch receptors. And so we'll get into some special senses a little later on to some of your sensory receptors a little later on in a chapter. But your touch receptors in the skin respond to some kind of mechanical stimulation. So when you touch something, that's actually opening an ion channel just because of mechanical stimulation. And so it's not really a voltage or chemical or ligand, but pressure or some kind of mechanical stimulation actually opens that type of gated channel. Sometimes temperature will affect ion channels as well. So sometimes temperature changes. That causes an ion channel to open. That causes an action potential. That sends a signal to the central nervous system to let you know that you're feeling that temperature change, that it's getting warmer. Now, the last section we'll look at, section 6, is on the synapse, and we've seen the synapse a little bit before, and I'm going to talk about muscle cells. This is just a word meaning a junction between two cells. That's what a synapse is. It's a junction. It's where two cells come together. Usually you have that's where you're going to find the action potentials, kind of bridging a gap. The gap is the synapse, and so a lot of times you'll have a nerve cell that's trying to send an action potential down through the axon, and then you has to cross over the synapse to get to the next cell. Many times it's a muscle cell or a gland cell or whatever kind of cell it is. And so that's what the synapse is, the junction where two cells come together and you're transmitting action potential, trying to get one action potential to cross over to the next one. A couple of vocab words here for you. Presynaptic cell, that's the cell that's actually sending the signal. And usually you're talking about like a nerve cell. A neuron would be a presynaptic cell. The postsynaptic cell, post means after, and so this is the cell that's receiving a signal. It could be a muscle cell, it could be a gland cell, it could be another neuron. So a couple of vocabulary words based on that information, based on synapses. If you have an axodendritic synapse, and it's telling you kind of what it's referring to, axo is referring to the axon or to the nerve cell, and dendritic is referring to the dendrites. And so this is a synapse or a junction between an axon of one neuron and a dendrite of a different neuron. So if you take a look at the diagram, this is figure 11.18. You've got a nerve cell here. It's got dendrites coming in, it's got axon coming out, and it has a couple of different axon ends, nerve ends of other neurons coming in. And so this one here has the axon coming down and is actually creating a junction with the dendrite. So in other words, it's sending a signal, its cell body would be up here somewhere, and it's sending a signal down to this neuron, and it's going through the dendrite connection. So that would be an axodendritic synapse. Axosomatics, soma is the word here for body or cell body. And so an axosomatic synapse just means that the axon is connected, forming a junction there with the cell body of the neuron. Then the axonic synapse, kind of hard to say, but that particular axon is actually just going down to another neuron. And so it's connection between the axon and another axon. So they actually kind of merge together. Most of the time when we've seen, like in the muscle physiology, we were looking at what would be called a chemical type synapse. And that just means that you have some type of chemical making the connection between the presynaptic cell, the axon, and the postsynaptic cell. And so in this case, you would have an action potential coming down the length of the membrane of that axon. It triggers voltage-gated calcium channels to open. Calcium would go inside of the axon or the presynaptic terminal, the end of the axon. That calcium causes these synaptic vesicles, which are storage vesicles that are storing up some neurotransmitters, to release their chemicals. Neurotransmitter may be like acetylcholine usually. That acetylcholine goes into the gap space, which is the synapse. It's a chemical synapse because you're dealing with acetylcholine. That chemical binds to the receptor, in this case a sodium-gated receptor on the postsynaptic membrane. It could be a muscle cell or a gland cell or whatever. It would be the postsynaptic membrane here. And that would open up the sodium gates. That sodium then would go inside the cell, and that creates a new action potential on that postsynaptic membrane. You also find electrical synapses. These are much faster, and you're going to find these mainly in cardiac muscle and then sometimes some smooth muscle as well. This time you don't have to generate a new action potential from a chemical or a chemical reaction. Instead, you have an action potential that's just carried through the cell through a gap junction or a connection between cells. And so, for example, if you see cardiac muscle cells all kind of lined up, they have what are called the intercalated disc area, and then they form gap junctions, which are just little pores or connections. And so you would have an action potential going down one cardiac muscle cell, and it can continue on, but can also kind of make a shortcut to go to the next cardiac muscle cell. And so this just allows for the connections to be much faster.